Hello everyone, I'm Dr. Cristabelle D, and I'm a postdoc fellow in the lab of Dr. Gerlinde Wernig, who is featured on the left here. And our research in our lab is focused on fibrosis, inflammation, and immune therapeutics. And today I'm going to talk about one of the projects in our lab, which is focused on dissecting the molecular mechanisms underlying the CGen CTNNB1 nexus that drives desmoid type fibromatosis. And fibrosis, while it's not a disease as sexy as cancer to study, is associated with more than 20 million fatalities in the United States alone, with a 5-year survival rate that is less than 50%. And this warrants the need for better molecular therapeutics that can increase the 5-year survival rate of patients. And with that in mind, and with the expertise that we have in terms of immune checkpoint therapeutics, we have a hypothesis that Jun, which has been discovered in our lab to play a role in modulating fibrosis, is essential for the proliferation of these primary patient desmoid tumors. And I will go through our rationale for how we designed our studies to arrive at the conclusions that we make in our research findings. So I'm preaching to the choir here, and everyone knows that our current understanding of DTFs is that they are associated or characterized by clonal proliferations, which are locally infiltrative, having a high recurrence rate. But most importantly, let's focus on the fact that they are associated with activated or mutated beta-catenin, which is a driving force of DTFs. So when we look at the transcriptional level of the pathways that are overexpressed in DTF patients, so we can see that the EMT pathways, cell migration, and extracellular matrix organization pathways, all are overexpressed in these patients or upregulated, and these are the pathways that are essential for the development of fibrosis. So we asked the next question as, since there are several key pathways enriched in DTF patients, and we know that beta-catenin is one of them and CGen is another one, can we elucidate and target this CGen beta-catenin nexus as a therapeutic target for the pathogenesis of DTF? And so, just to give you a brief introduction of CGen, for those of you who don't know, it is a master transcriptional regulator which induces the expression of several of its target genes to promote proliferation, migration, immune checkpoint, ultimately promoting survival. So when we looked at the transcriptional targets that were upregulated in DTF patients, we found that there were 92 gene targets that were not only upregulated in DTF patients, but were also downstream targets of CGen, which led us to ask the question if we could see activated GEN and beta-catenin expression and if this could be correlated in patient samples. So when we looked at patients, we found that phospho-GEN, which is the activated form of GEN, and beta-catenin both have a high expression in DTF patients. And performing immunoblotting, we found that this expression in desmoid patients was very high. And performing immunoprecipitation studies with a pull down of CTNNB1, which is beta-catenin, and GEN, we found that GEN and beta-catenin interact with one another at the molecular level. So then we asked the question, if we knock out GEN, what would be the effect on beta-catenin activity? And here we see that with an efficient knockout of GEN using our CRISPR system in primary patient desmoid cells, we show that, not surprisingly, there is an inhibition of growth in the cells, which is measured by EDU labeling. But very surprisingly and with showing great promise is that the knockout of GEN actually significantly alters the beta-catenin activity and decreases it. So now we know that not only is CGen and beta-catenin interacting with one another, but they are But C-Jun also regulates the activity of beta-catenin. But would this translate into an effect in vivo? And so here we see that when we ectopically transplanted primary cells that had Jun overexpression, we found that Jun overexpression significantly led to a survival of the ectopically transplanted grafts in our novel kidney capsule model that is established in our lab. And so with these very promising results, we then asked if we could recapitulate the patient phenotype in in a genetically Jun-inducible mouse model. And so on the left panel here, you can see that we have our construct that we use to generate these mice. And after inducing inducing doxycycline intraperitoneally, we see that there is a Jun induction. And this Jun induction correlates with desmoid-like lesions which recapitulate the phenotype that is seen in patients. And we can see the activated Jun and Ki67, which indicates increased cellular growth. So our very promising results indicate that in summary, C-Jun is definitely an essential promoter of the pathogenesis of DTF. It functions via its interactions with beta-catenin. Overexpression of Jun in desmoid cells promotes a diseased phenotype in vivo, and using our novel inducible model, we can recapitulate the pathological features of human DTF in vivo. But then we ask the question if we can safely target the Jun or Wnt pathways for DTF treatment, because since Wnt and Jun pathways are essential for developmental cellular processes as well as for tissue homeostasis, would the side effects be abrogated if we target something that is downstream of Jun? And so coming back to one of the downstream targets that Jun regulates, we know that CD47, which is an immune checkpoint marker or a don't eat me signal that inhibits phagocytosis in cancer cells, we show that C-Jun directly regulates CD47. And in our prior published ATAC-seq data, we show that in primary fibroblasts, as you can see here, C-Jun affects the promoter occupancy at the CD47 locus, thus indicating that C-Jun dynamically controls the chromosomal accessibility of CD47 and drives CD47 transcription. So now that we have the mechanism, to put it very simply, we have CD47 as a potential strategy to treat DTF by activating the innate immune system. And how we would do it is, in the simplified model, you can see that in the presence of CD47, an activated fibroblast would not be phagocytosed. But with our antibody, which is currently in clinic, you could block CD47 and activate the innate immune system to induce phagocytosis of the pathogenic fibroblasts. And so in sum, with these promising results that we have, we would now want to have a future direction in studying the effect of novel inhibitors that can be Wnt inhibitors or inhibitors like tegavibint in combination with the blockade of CD47 for the treatment of desmoid tumors, both validated in vitro and in vivo. And we also want to understand the transcriptional landscape of Jun-mediated targets, because we already know that there are 92 targets that are downstream of Jun and are also significantly overexpressed in DTF. And we want to understand in the fibroblast how Jun regulates these different targets, and we would like to perform mechanistic studies. We currently have primary human desmoid cells as a kind gift from the Broad Institute, and we want to test with the mechanisms and our treatments in combination to arrive at the best possible solution to solve the current challenge that we have with treating a disease like DTF. So with that, I would like to acknowledge my lab members, Dr. Tristan Loeb, who has done most of the work in the study. He spearheaded this project. Of course, my PI, Dr. Gerlinde Wernig, who I mentioned in the first slide, my co-postdocs, Dr. Chue and Deng, and Dr. Liu, who is a collaborator and a bioinformatics specialist from another lab. And definitely no research can be completed without the support and encouragement from organizations like the DTRF. So we definitely want to thank the Desmoid Tumor Research Foundation for their continued support, and hope that we continue to get this kind of support so we can make greater strides in our research. With that, we can take any questions. Thank you.